Right, these were the components of the JG apparatus. Now finally, I need to explain one more cell which is not a part of this JG apparatus. We have seen already. So fine, I need to explain just one more cell which is not a part of it. Type 1 cortical interstitial cell. Around the peritubular capillaries in the renal cortex. So in the cortex, around the capillaries, so around the peritubular capillaries, but outside the nephron, there is this interstitial cell. So it's called as cortical interstitial cell. This cell synthesizes erythropoietin. It's not a part of JG apparatus, so don't confuse. JG apparatus had 3 cells. We are done with that part. I am just adding it as an additional information. Erythropoietin is synthesized and secreted by this particular cell. It has oxygen sensor. Just as the carotid bodies, chemoreceptors have oxygen sensors, the O2 sensitive potassium channels. Just as pulmonary vascular smooth muscle cells have oxygen sensors, hypoxia sensors, O2 sensitive potassium channels. So even here, because then it has to sense the hypoxia and then release the erythropoietin. So erythropoietin is synthesized by the kidney, but from where? From here. Right, so that was another component of the nephron. Before we proceed any further, I just mentioned to you some time back that we will see the glomerular filtration barrier. We will discuss it again in the context of GFR. But since we are still in the introductory part, let's just quickly go through it. Look, the structure of the glomerulus. Glomerulus is a tuft of capillaries. Fine. The blood comes into the glomerulus via afferent arteriole, and then the glomerular capillaries will filter the substances. But some of the substances cannot be filtered out from these capillaries into the Bowman's capsule. So, which substances do not get filtered at the glomerulus? And why is that? Well, that is because of the so-called filtration barrier. Filtration barrier. Let's therefore see the filtration barrier. Here is a glomerulus. Here is a glomerular capillary. Again, a diagrammatic representation. Glomerular capillary. Well, there is outside these capillaries, there are epithelial cells which are called as podocytes. So here are the podocytes, and they have certain foot processes. You will see that these are the foot processes of the podocytes. Okay. So basically we will have 3 components that constitute the glomerular filtration barrier. It will create a barrier for the filtration of substances. Those substances will not be filtered out. Therefore, let's make this part enlarged. Just a small part of the glomerular capillary and outside will be enlarged. Here is a capillary. First part of the filtration barrier is the endothelial lining of these capillaries. As you can see, the blood vessels are lined with the endothelial cells. So, endothelial lining of the capillaries. The second component is the basement membrane. The basement membrane and all its glycocalyx and all the components. And Then outside this basement membrane, there are those podocytes, the epithelial cells, the podocytes. Okay. So blood will come into the capillary, glomerular capillary. Now the filtration will start, but there is a barrier, endothelial cell, the basement membrane. Let me add, basement membrane and that glycocalyx has the negative charges. So please keep this in mind, the basement membrane has the negative charges, right? And then outside that there will be those podocytes with their food processes. There is also the slit diaphragm. The slit diaphragm. So let me just show it diagrammatically. The slit diaphragm. Components of the slit diaphragm include the nephrin, the podocin, and other such components. So now the filtration will occur through these pores in the capillary basement membrane. Filtration will occur like this. The substances will be filtered out. But one thing, cells do not get filtered. Okay, so that's out of question. Plasma is supposed to be filtered. Okay. Now in the plasma, what all substances are not filtered? The proteins. And therefore, proteins, why don't they get filtered is because of two reasons. One is proteins are anionic and proteins are large molecules, macromolecules. So remember, because they are anionic, they will be repelled by those negative charges on the basement membrane. So they will not be attracted, they will not be pulled for filtration. They will be repelled and they will go in the efferent arteriole. That is what we saw just now in the glomerular-tubular balance. So that's one. And the second is large molecules. Large molecules are not able to cross these pores and the filtration slit diaphragm. So please remember, anionic macromolecules are the last one to be filtered. I mean, they will not be filtered at all. Micromolecules and cationic molecules, they are filtered rapidly. So keep this in mind. Okay. That's how the proteins are not filtered because of this filtration barrier. So that's one thing that I wanted to add in the context of the filtration and the filtration barrier. You will learn, like for example, nephrotic syndrome. Nephrotic syndrome is going to be resulting from damage to this filtration slit diaphragm. I told you just now there are certain proteins like nephrin, podocin, etc. So they are damaged, and therefore what happens is the proteins do get filtered out and proteins are lost into the urine. I mean, those proteins are damaged plus the negative charges, but there is a damage to the basement membrane as well, and therefore proteins do get filtered out and lost into the urine in the conditions such as nephrotic syndrome. But otherwise, normally they don't get filtered because of this reason. Right. So that was the introductory part related to the nephron. Now the next part is we will see the renal circulation, and that will finish our first section of the discussion, of the kidney discussion. Let's see the renal circulation. I am not going to describe the anatomy of it. Already, if I were to speak of the anatomy, from anterior to posterior, how the things enter or leave. If this is the kidney, there is artery that goes in, vein that comes out, and there is ureter that comes out. So, how they are situated anterior to posterior is veins, the renal vein, then artery and ureter. Vein, artery, ureter. Vein, artery, ureter. Their anatomical relation from anterior to posterior. And then renal artery, then it enters and then breaks into arterioles, then there are afferent arterioles, then glomerular capillaries, then efferent arteriole that breaks into peritubular capillaries. So, and for just I'll add one more thing. In the case of those long loops of the juxtamedullary nephrons, the blood vessels called as vasa recta with similar long loops are present in the medulla. We will discuss about them later when we discuss concentration of urine. So, in the renal circulation, we are going to discuss the peculiarities of renal circulation, functional peculiarities of renal circulation. Let's see. First, renal circulation shows auto-regulation of blood flow, right? Three vital organs, brain, heart, kidney, they will show auto-regulation of blood flow. So, kidney shows auto-regulation of blood flow. Second point, this is the only circulation where capillaries lead to arterioles. Capillaries lead to arterioles, isn't it? Glomerular capillaries lead to efferent arteriole. That's exceptional because everywhere else in the body, arterioles lead to capillary and capillary leads to veins, venous compartment. But that does not happen here, at least in the glomerulus, in the nephron part. Okay, next, very high blood flow. That again is a very peculiar feature. Kidneys have exceptional amount of blood flow. It's about 22 to 25% of the cardiac output. So, that will be second best. Highest is the liver. We've seen that already. Liver is 25 to 27% of the cardiac output. But kidney weighs only 1 to 2% in the body and it receives 25% of the cardiac output. So, it's a disproportionately high fraction of the cardiac output, right? Remember this in the next exam also, in any entrance exam, whether it's next or whether any type of exam, physiology also bears disproportionately higher amount of importance. Why? Because direct questions will be there from physiology. That apart, every other subject of the clinical importance will need understanding of physiology. So, don't take it as just okay, a few questions likely to be asked. No, if you want to answer the medicine questions, you better understand the physiology behind it. Otherwise, problem solving questions become a problem. But I am sure with these videos that problem is sorted. Don't you worry at all. Okay, so coming back to the topic at hand, 22 to 25% of the cardiac output, very high. Just 2% of the weight of the body and 25% of cardiac output. Also, blood flow per 100 gram of tissue per minute, that also is very high. Now naturally, because small organ and massive blood supply. So, 400 ml per 100 gram of tissue per minute, that's the blood flow per 100 gram of tissue per minute in the kidney. that is also second best. If you recall, if you have done CVS by now, tell me which is the first. Carotid body. Yes, carotid body receives 2000 ml per 100 gram of tissue per minute. So please remember, if they ask you which tissue in the body receives maximum blood flow per 100 gram of tissue, the answer would be carotid body. But if they ask you which organ receives maximum amount of blood flow per 100 gram per minute, organ, kidney, tissue, carotid body. Okay, but anyways, the point was very high blood flow among the top two in the body. Next point, capillary hydrostatic pressure. Capillary hydrostatic pressure. If you remember, elsewhere in the body, in the systemic circulation, please recall the CVS chapter, we have seen this, that at the start of the capillaries the pressure is 30 mm of Hg, by the end of the capillaries the pressure is 10 mm of Hg, and therefore average capillary pressure is 15 to 20 mm of Hg, right? That's the average capillary pressure elsewhere in the body. In the kidney, the pressure in the glomerular capillaries is 3 to 4 times this pressure. 3 to 4 times. So, capillary hydrostatic pressure is highest in the body in the glomerular capillaries. How much is it? Look, different figures are given in different books. It is 60 mm of Hg. So, just remember like this, elsewhere it is 15 to 20, but in the glomerular capillaries, it's 3 to 4 times that pressure. So, it's 60. And why is that? The reason is also very obvious, because afferent arteriole is little wide and the efferent arteriole, which takes the blood out of the glomerulus, that is little narrow. So, the amount of blood coming into the glomerulus per unit time, does not all of it leave the glomerulus per unit time. Which means what? Which means the blood accumulates in the glomerular capillaries. And you remember we have discussed the Starling's equilibrium when I told you that greater the amount of fluid accumulated, greater is the hydrostatic pressure. Okay. So, therefore, it's highest in the body, the hydrostatic pressure is in the glomerular capillaries. Next point, fifth point, capillary filtration coefficient. Capillary filtration coefficient. You know, everywhere in the body, the capillaries filter out the substances, water and the substances. And this occurs with a certain rate. And that rate of filtration is called as capillary filtration coefficient. This also is highest in the body in the glomerular capillaries. So, another highest thing. How much is it? It is 12.5 ml per minute per mm of Hg, very, very high. Elsewhere, it's just in less than 1 ml per minute per mm of Hg in the systemic capillaries, in the tissues. Here, it's very high. Alright. So, 12.5 ml per minute per mm of Hg. I need you to remember this, because when we start the discussion on GFR, I am going to ask you this once again to recall this. I will answer it, but then remember this. 12.5 ml per minute. So, you know how to read the coefficients. Okay. For every 1 mm of Hg of filtration pressure, in 1 minute, 12.5 ml fluid is formed at the glomerulus, means 12.5 ml filtrate is formed by the glomerular capillaries. I mean, if the filtration pressure is 2 mm of Hg, then in 1 minute 25 ml filtrate will be formed at the glomerulus. That's how you read it. Then next, sixth, arteriovenous oxygen difference. Arteriovenous oxygen difference. This is least in the kidneys. Least 12.5%. So what is this arteriovenous oxygen difference? Look, elsewhere in the body, when 20 ml of oxygen is carried by every 100 ml of arterial blood, we have done this in the respiratory system as well. When this blood goes into the tissues, out of this 20 ml, generally the tissues extract 5 ml of oxygen. Which means what? Which means the remaining 15 ml goes back into the vein, venous blood. And so, arteriovenous oxygen difference is 5 ml, 20, 15, 5. So, 5 out of 20, 5 on 20 is 25%. That's the AVO2 difference elsewhere in the body, okay. This is 75% in the case of heart, so highest there. We have done this in the coronary circulation. And this is least in the kidney. It is just 10 to 12.5%, least in the kidney. The reason is also obvious. The reason is small organ but massive blood supply. So, massive oxygen is coming in, and therefore, whatever it takes out, it will be less percent only in the context of the blood that is coming in. We saw that already, 2% weight and 25% cardiac output going into it. And so, how much soever oxygen it extracts, it will be less in the context of that massive blood flow and massive oxygen delivery. And therefore, it's the least in the kidney, 10 to 12.5%. Right. So, another unique feature in the kidney. Right. So, next, seventh, another unique feature. This will be the last one. Elsewhere in the body, now listen to this part, again very amazing. Elsewhere in the body, metabolic rate in that organ or tissue regulates blood flow to that tissue or organ, right? Increased metabolic rate. I told you some time back about the Bernays hypothesis. We saw Bernays hypothesis. Increased metabolic rate, increased ATP breakdown, so increased adenosine formation, and that will dilate the vessels, the blood flow will increase. So, metabolic rate regulates the blood flow to that organ, any organ. There are only two exceptions to this, only two exceptions. One is the skin, cutaneous circulation. Cutaneous circulation is not regulated by the metabolic rate of the skin or something. It is regulated by the temperature. The primary regulator of cutaneous blood flow is the temperature, ambient temperature. And the second exception is kidney. In the kidney, it is exactly opposite. In the kidney, blood flow regulates the metabolic rate. Do you get the point? Elsewhere, metabolic rate regulates the blood flow. The kidney is exactly opposite. Blood flow regulates the metabolic rate. How? Very obvious. Increased blood flow means increased GFR. Increased GFR means increased sodium filtration at the glomerulus, which means increased tubular load for sodium reabsorption. Increased tubular load for sodium reabsorption, isn't it? And you saw, we have already seen this some time back, that the metabolic rate is directly linked to what? Is directly linked to the rate of sodium reabsorption. So, increased tubular load for sodium reabsorption means there will be increased metabolic rate. So, please remember, the metabolic rate, oxygen consumption, ATP breakdown is regulated by the blood flow. Okay. Elsewhere it's the other way around. Metabolic rate of the organ regulates the blood flow to that organ. In the kidney, blood flow to the kidney regulates the metabolic rate in the kidney. Very exceptional. Right. So, these were the peculiarities of the renal circulation. That was the first section on the excretory system.